Assalamu alaikum. My name is Sarah Shaheen and I'm from Biotechnology B 2015 and my group members are Nayab Said, Kamal Sanaullah and Samiabad. Here we are going to demonstrate the DNA extraction from dates, and the dates which we have used are Ajwa. First of all, I will demonstrate you all about the equipments and the material required. Firstly, we require sodium chloride as well as the ethanol, absolute alcohol, distilled water. A detergent is used, as well as a gauze piece or a filter paper is required along with a stirrer or a spoon, a tripod stand, some beakers, test tubes, as well as Bunsen burner. This is the extract which we have prepared by boiling the dates. Firstly, we incubated the dates for 3 to 4 days, and after that, after the incubation, we brought them out from the incubator, and after that, we boiled it in hot water, not way more than 100 degrees centigrade, because of that, the proteins and the contents of the dates would degrade. So we carefully boiled the dates as well as mashed them with hands, and this was the extract which we obtained. After the extraction of this extract, we then filtered the extract using this gauze piece or a filter paper. As these are dates, so we used this gauze piece as it was very easily available, so the extract would easily flow through it. Now, in order to filter the extract, we have used this gauze paper, and now we will carefully filter this extract. With its filtration by the gauze piece, the filtrate will pass through the gauze piece and gradually it will pass into the next beaker washed with the distilled water. We have washed it with distilled water in order to avoid contamination, as we have obtained the filtrate. Now, as we have the filtrate, we will use this detergent. This detergent is used in 1 to 2 drops. It is poured 1 to 2 drops of the detergent, and it is then mixed into the filtrate and the extract obtained. Here we are mixing the detergent. And now we have taken distilled water and we will heat this water on a hot Bunsen burner flame. In order to heat this water, we will add sodium chloride in it. With the addition of a few spoons of sodium chloride, the solution will become salty. Now here we are about to add sodium chloride in it. A minute amount of sodium chloride is added in order to make a salty solution. This is how the salt is added, and now the boiled water is taken, and this boiled water is added into the filtrate we have obtained, where there the detergent is being mixed. As this hot water is added, the boiling volume is made up to 200, and then we will mix this filtrate with this hot water. And after that, when this hot water is added, again it is subjected to filtration, because being the dates, there will be high amount of material which is present in solid form. So in order to again get a pleasurable and the bestest filtrate, a homogeneous mixture, we will again undergo the process of filtration. Here we have again used a gauze piece, and the solution is being filtered again in order to get the required and minute amount of the filtrate. As you can all see that there is a clear solution formation, and this clear solution gives us a complete and a homogeneous mixture which can be used for the extraction of DNA from dates. As we all know that the precautions and safety measures are to be taken in a laboratory while working in a laboratory, so we should first do is that filling this test tube with the extract we have obtained, the homogeneous mixture. When this is filled to a required amount, after the filling of the test tube, we will add then the absolute or the ethanol alcohol. This alcohol is added so that the DNA threads obtained might rise up to the surface, and it should be added very slowly because in order to obtain the clear threads of DNA, the alcohol's surface or the layer should be up on the upside above the surface of the extract the homogeneous mixture obtained. So let's do it now. Moving towards the next step. Now, we have taken the alcohol in a small beaker in order to avoid any sort of contamination. So it is poured very slowly in order to obtain a layer on the upside of the test tube. A layer is obtained on the upper surface, and gradually we will cover it with a silver paper. When it is covered with a silver paper, then the vapors will rise and contamination will be avoided. So let's wait for a while and cover it with a silver paper, and after a while we will see the DNA threads in the extract obtained from dates. As you can all have a check at it clearly that we have covered with a silver paper, and we have seen our test tube after half an hour, and clear threads of DNA can be visualized in it. These are the threads of DNA which we have extracted from Ajwa. And these threads actually signify our experiment to be successful. And this is how we extracted DNA from dates. Thank you so much.